Welcome back to What Did I Do in Lab Yesterday, where I tell you guys what I did in my biochem lab, last week actually. So last week was week 1 of crime scene investigation. No, not a real crime scene, a fake one. What it basically was just a fun way to do DNA analysis. So what happened, we had a few samples of DNA. There was one that was found on the crime scene, a blood sample, and also a not cheek, but skin sample. And then there was blood samples from 4 different suspects in the murder case. So what we did first, we had the one sample and we had to run it on gel electrophoresis, and basically gel electrophoresis is used to separate DNA based on fragment size after it's been digested by restriction enzyme. So once it's all digested, you run it through the gel electrophoresis machine and you get a gel. So this is the gel that we ended up with. The first row is the ladder, which is basically just used to compare DNA fragment sizes. The first row is the DNA sample that was found on the murder victim, and then the last 4 rows are the DNA samples from each of the suspects. As you can see here, the row with suspect 3 matches the DNA samples in the first row, so it's looking pretty likely that we've got suspect 3 as the murderer here. I just got distracted because I needed to go print off recipes for my roommate. I don't remember what I was talking about, but anyway, we had to wait for the gel electrophoresis to do its whole thing. But while we were waiting for that, we had a bunch of other stuff to do. We had to get the other 2 DNA samples ready, and so those 2 DNA samples were from other cell types. I think one was like a skin sample, so we had to do stuff with that because there wasn't actual enough DNA to put through the gel electrophoresis. So we did that by sticking it through PCR, or polymerase chain reaction, which is basically used to take a small amount of DNA and amplify it so there's a lot more DNA available to use. So that's a whole cool process, but we had to prepare that, leave it to just do things, get the DNA prepared to be amplified, now stick it into the PCR machine to leave it to cook overnight to get all the DNA that we needed. So we didn't actually have enough time to deal with the DNA we put in the PCR, so that's what we're going to be doing this week in the lab. Take that DNA and run it through more gel electrophoresis and confirm the murder.